Hi, my name is Brianna Shaw, and I'm going to do a presentation on mitochondrial gene editing. In relatively recent years, genome editing technologies have been on the rise. These technologies act like scissors, cutting the DNA at the specific spot. Scientists can then use a joining system to remove, add, or replace the DNA where it was cut. The most famous gene editing tool currently is CRISPR-Cas9. Scientists developed a guide RNA which finds the spot in the nucleus in the cell where editing activity should take place. Then, the guide RNA attracts Cas9 to this location and to bring it to where it needs to be cut. Once the Cas9 snips the DNA at the spot, a break is created in both strands of the DNA. Then, two things can happen. Either the cell will work to fix the break on its own because it can identify that there's a breakage in the DNA, and it'll do this by cleaving the two DNA sites back together. Or it will be corrected by a replacement segment of DNA or a whole new gene altogether provided by scientists in a lab. While CRISPR-Cas9 is a powerful tool for cleaving double-stranded DNA, it is not able to perform the same function with mitochondrial DNA. Inherited or acquired mutations in mitochondrial DNA, also known as mtDNA, are associated with a wide variety of human diseases. Mutations in mtDNA cause over 150 distinct syndromes, and each year, US children are born with no cure for these diseases, and approximately 1 to 4,000 of them are born with these diseases. Tools for introducing precise modifications into the mtDNA are needed to both understand the pathways and to potentially treat these diseases. There wasn't much progress until very recently. Scientists at the University of Washington in Seattle identified a toxin secreted by the bacteria Burkholderia sinusospacea, which surprisingly proved to be the solution needed to create a mitochondria-friendly base editor. This toxin killed bacteria by causing destructive DNA mutations. Unlike all other converting proteins, the toxin could directly make changes to a double-stranded DNA instead of first having to convert it to a single-stranded DNA. It worked by attaching to DNA and then modifying one letter cytosine so that it resembled a different one. In our case, thymine. These intentional DNA typos were what brought down the toxin's victims, aka the mitochondrial DNA. Upon doing more research to determine what was happening at the molecular level, the scientists realized that this conversion was as lethal to mammalian cells as it was to bacterial targets. So, in order to modify the toxin so that it didn't ruin the double stranded DNA, researchers split the protein into two non toxic halves, where the two pieces changed cytosine to thymine only when they were brought together to the same spot of DNA. They then attached short pieces of proteins called tail proteins to the end so then they could be chosen to target specific stretches of DNA. In further cell culture experiments, researchers saw the mitochondrial editor successfully converted cytosine to thymine at the intended mtDNA locations, with ranging efficiency, however. While this may seem minor, it has major implications. So far, base editors have used proteins like Cas9 to open target DNA into single strands before making a change. However, pieces of RNA required for the function of these proteins can't get into the mitochondria. This discovery was a major feat. A base editor that works on double stranded DNA was found and would no longer need to depend on Cas9. The future will aim to look to improve efficiency, develop new types of mitochondrial editors that can produce other bases. So instead of producing only thymine, in this case from cytosine to thymine, you could go from, and like this example shows here, you can go from cytosine to guanine, adenine to thymine, adenine to guanine, guanine to cytosine, guanine to thymine, etc. Also, we need to determine if it works on animals and then potentially humans after that. While this is just a first step, it is revolutionary in the aspect of gene editing of mitochondria and beyond. This, this is what the research article was about. So while this is only the beginning stages of finding something new, many scientists are amazed at the feat of this because They have never been able to really look at the molecular level into these diseases and into these syndromes as to what is happening and how there could be a cure, even for a drug, to solve any of these problems or symptoms.